Greetings, I'm Mason Brooks, Managing Editor of Epigene, and welcome to today's webinar presented by Dr. Brian Egan titled Bringing ChIP-Seq Data into Focus, Advanced Normalization Strategies. Dr. Egan is the Epigenetics Services Manager at Active Motif, and through his daily hands-on experience, he and his colleagues have developed some very useful background normalization strategies for handling ChIP-Seq data, which I'll be sharing more with you today. Before I hand things over to Dr. Egan, I'd like to thank Active Motif for their support and sponsorship of this webinar. Active Motif is committed to enabling researchers who investigate chromatin biology and epigenetics through the development of high-quality products, including chromatin IP kits and reagents, high-quality antibodies to histones and chromatin proteins, kits to analyze DNA damage, DNA methylation, and histone modifications, and of course, ChIP-Seq services. So please click over to their site and check those out when you can. One last note before we begin, we'll have a question and answer session after the presentation, so please type any questions that you might have into the questions box. This will appear on the right-hand side of your screen, and I'll be sure to pose those after the talk. So with that out of the way, I'll turn the floor over to Dr. Egan.
Thank you, Mason, and thank you to everyone who's joining us today. My name is Brian Egan, and I lead the Epigenetics Services team at Active Motif in Carlsbad, California. For those of you who don't know, beyond being a kits, antibody, and reagents company, Active Motif offers end-to-end ChIP-Seq services. And because we do so much ChIP-Seq, it gives us a unique opportunity to identify problems and challenges in the field, and it allows us to team up with other researchers to solve these problems. So today I'm going to talk about a persistent problem we've seen for the last couple of years and what we've done to try and solve that problem. And that problem is that we do not see the expected changes in H3K27 trimethylation in our ChIP-Seq data after the addition of EZH2 inhibitors. Before I present that work, I want to get everyone up to speed by giving a brief introduction to epigenetics and ChIP-Seq, and then I'll talk a little bit about our ChIP-Seq service, and the majority of the slides will cover the spike-in strategy that we developed to normalize ChIP-Seq data. Epigenetics refers to the functionally relevant and heritable modifications that occur on the genome that are not related to changes in nucleotide sequence. These modifications can occur directly on the genome in the form of methylation of cytosines, but can also occur on histone tails. There have been a number of histone modifications identified, but the most widely studied are methylation and acetylation. These modifications affect the openness or compaction state of the chromatin and eventually lead to changes in gene regulation. There are three main groups of proteins that regulate these modifications. They're loosely referred to the writers, erasers, and readers. The writers are enzymes that deposit these modifications. The erasers are enzymes that remove the modifications. The readers are a group of proteins that do not have enzymatic activity, but function through binding certain modifications and recruiting other proteins to chromatin. I think it's important to note that like inducible transcription factor binding, changes in histone modifications are inducible and can be the endpoints of intracellular signaling cascades. To illustrate this point, I'm showing some ChIP-Seq data that we generated, and I've just zoomed in to a single gene, and we show that after interferon gamma treatment, we can recruit STAT1 to the promoter of this gene, but at the same time, we also induce H3K4 trimethylation modification at that promoter. So there's a very intricate interplay between both transcription factor binding and histone modifications that regulate gene expression. Obviously, the field of epigenetics has grown tremendously over the past 10 years. And we've reached a point now where there's a lot of interest in exploring epigenetic marks and modifying proteins as biomarkers or drug targets. Epigenetic modifying enzymes, and now even the reader proteins, are hot drug targets, and pharmaceutical companies are making significant investment developing drugs that target these types of proteins. And right now, probably the most well-known or the most popular targets in pharma are against the methyltransferase EZH2 and the reader protein BRD4. As of today, there are currently 4 drugs on the market that have epigenetic modes of action. 2 of those are DNMT inhibitors, and 2 of those are HDAC inhibitors. And you can see by this list that there are a number of other inhibitors at various stages of clinical development. I think it's safe to say that most, if not all, large pharmaceutical companies now have programs dedicated to developing inhibitors against epigenetic modifying proteins. And most of that effort is occurring in the field of oncology. But there's also a number of smaller pharmaceutical companies that are solely dedicated to developing inhibitors against these types of proteins. I've highlighted two companies in this list, Genentech and Constellation Pharmaceuticals, and the reason for that is that both of these groups collaborated with us on our project to normalize ChIP-Seq data. One of the main techniques used to detect histone modifications as well as transcription factor binding at specific genes or across the genome is chromatin immunoprecipitation, or ChIP. In this technique, living cells or animal tissues are fixed with formaldehyde. Formaldehyde is a small molecule. It readily permeates the cell membrane, resulting in protein-protein and protein-DNA cross-linking. Once the cells are fixed, the cells are sonicated to lyse the cells and to fragment the DNA. The DNA is typically about 500 base pairs long, with the native proteins covalently attached. That chromatin can be used in immunoprecipitations using an antibody against the target of interest. So you're enriched for that target, but you also enriched for the DNA to which that target's bound. Once you reverse the cross-links and get rid of the protein, you have a population of DNA that is representative of all the binding sites across the genome. And that ChIP DNA can be analyzed in different ways. The first on a gene-by-gene basis using qPCR, but also the whole population can be sequenced and analyzed bioinformatically to identify where those binding sites are. As I mentioned at the beginning, Active Motif has an Epigenetics Services group that performs end-to-end ChIP-Seq, and Active Motif's ChIP technology follows the same basic steps that I outlined in the previous slide. However, over the last 10 years, we've developed our protocols to give optimal signal, but probably more importantly, minimal background. Additionally, we've added a number of quality control steps. So our goal is by the time we get to sequencing, we want to have complete confidence that we're going to get a good ChIP-Seq data set. So customers typically send us fixed cells or frozen tissues. We prepare the chromatin, and as our first quality control, we quantitate the chromatin that comes from each reaction. If we're going to do multi-sample experiments, we want to be sure to add the same amount of chromatin into every reaction. We make recommendations for the antibodies and perform the ChIP, followed by qPCR at positive and negative control sites. If the qPCR looks good, we proceed to library generation. We QC the libraries in 3 different ways. We then do the sequencing on an Illumina platform and analyze the data. Active Motif's Epigenetics Services Group actually started as a different company back in 2002 called GenPathway. We recognized that chromatin immunoprecipitation was going to be a valuable technique and just started developing our protocols. In 2004, we did our first paid ChIP qPCR assay. We now generated more than 3,000 ChIP-Seq data sets, which is certainly more than any other commercial group. We've successfully performed ChIP using antibodies to more than 220 different targets. We've processed over 12,000 samples for ChIP in that time frame, and we've prepped chromatin and done successful ChIP on over 15 different species and 25 different cell types. And because of the nature of the services business, we have had the privilege of working with a lot of really great scientists over the years, and much of that work has ended up being published. So these are just a few examples of our more recent publications where our work has appeared. And as a scientist who's worked in the field of epigenetics for quite some time now, it's exciting to see epigenetics migrating into other fields. So I think epigenetics is becoming a core discipline within gene regulation and is starting to move into other areas. You'll see there's a few papers here related to oncology. That's no surprise. A lot of the work in epigenetics is focused on cancer. But we also see things like circadian rhythms, inherited behaviors with epigenetic roots, as well as immunology. There's one more topic that I want to cover today before I present the spike and normalization strategy that we've been working on. And that topic is how good is ChIP-Seq as a technique at detecting differences across multiple samples. I've talked to many people about ChIP-Seq, and I've heard a lot of people say that ChIP-Seq is good for detecting binding versus no binding, but it's not necessarily good for quantitating or detecting degrees of binding. So I think it's widely accepted that you can detect on-off situations. I think a good model for this is in stem cells. So as the cells differentiate, you can clearly see both activating marks and repressive marks shifting during that differentiation. The story is not so clear when it comes to detecting degrees of changes at commonly bound sites across multiple samples. And since many people have seemed to have problems detecting these changes, there's been a number of publications over the last few years trying to address this problem bioinformatically. And personally, I'm not so sure that these bioinformatic approaches make huge differences to the data sets. Maybe for low-quality ChIP data sets, you can improve them by using these types of techniques. But I'm of the opinion that if you're doing ChIP well, you should be able to detect degrees of binding across multiple samples. Of course, this may be influenced by cell type and antibody, but I think in general it can be done. So here are a few examples of those types of studies that we have performed. So in this example, we were looking at a specific histone modification in the presence of an inhibitor that should lead to a decrease in that modification. So without the inhibitor, we see the mark at a few genes here, and as we increase the concentration, we see that mark declining. The entire data sets for this project looked very similar. I've just zoomed in on a few areas to illustrate the point that you can see degrees of binding. The same is true when looking at transcription factors. We've had many assays where we've induced transcription factors over time and have seen those changes. Here's an example of an assay where we looked at estrogen receptor binding after estrogen treatment. So without the treatment, we do see a little bit of background binding for estrogen receptor, but at 30 minutes, that signal goes up. It continues to go up at 1 hour and 6 hours. I think these are good illustrations of how you can detect degrees of binding and not necessarily just on-off situations with ChIP-Seq. However, in the project I'm going to talk about today, we did not see these differences. And that's what led us down the road of developing a spike-in system that allows us to see these differences. In that project, we did a ChIP-Seq in the presence of EZH2 inhibitors, and we were not detecting the expected decrease in H3K27 trimethylation occupancy. EZH2 is a methyltransferase. It methylates H3K27. EZH2 is also a cancer target. It's frequently overexpressed in cancer, and there are some cancers that have activating mutations. And these include diffuse large B-cell lymphomas, follicular lymphomas, and melanomas. As such, there's a lot of interest from pharmaceutical companies to develop inhibitors to EZH2 with the idea that you could bring K27 trimethylation levels back down to something that is closer to normal, and this would have some therapeutic benefit. So there are a lot of groups developing EZH2 inhibitors and characterizing them, and some of the questions they're asking are how do these inhibitors influence global K27 trimethylation patterns? How do these epigenetic changes influence gene regulation? And how do these inhibitors influence clinical outcome? The question we can contribute to with ChIP-Seq is how do EZH2 inhibitors change global H3K27 trimethylation patterns? We've done a number of K27 trimethyl ChIP-Seq projects over the years using a number of EZH2 inhibitors. And in every single case, we have not been able to see the expected decrease in K27 trimethylation. So here's a figure that encompasses 4 different experiments using 4 different EZH2 inhibitors. And you can see in each case, in the presence of the inhibitor, we're not seeing a decrease in K27 trimethylation. So this is a big problem for the people developing the inhibitors because they've shown in biochemical assays that the inhibitors actually do inhibit. And they've also seen global decreases in K27 trimethylation as measured by mass spec in Western blot. So once we identified this problem, we were lucky enough to team up with 2 different groups, the one at Genentech, led by Marie Classon, and the one at Constellation, led by Patrick Choyer. And so here we have some data from those groups. This is the Western blot showing that the inhibitor used by Genentech, which is GSK126, results in a global reduction in K27 trimethylation levels, and at the same time, a slight increase in K27 acetylation levels. And we'll come back to this again in the coming slides. In the Constellation samples, one of their compounds that they're using in this particular experiment is CPI-360. So by Western blot, you can detect global K27 trimethylation levels. They're decreased at 2 days after treatment and significantly decreased at 4 days and 8 days. So the question is, why do we not see these expected decreases in the ChIP-Seq data? Is it a function of the ChIP reaction? That is, are those differences present in the ChIP DNA? Or is it a function of the downstream processing and preparation for sequencing? So the answer to the first question is yes. We actually can detect those differences in the ChIP reaction. So here's 3 separate experiments using 3 different EZH2 inhibitors. We performed ChIP followed by qPCR at select genomic loci. And in each case, you can see somewhere between a 2- and 4-fold decrease in K27 occupancy in the presence of the inhibitor. As I mentioned, we've done many other inhibitors. Here's an example of an experiment where we did BRD4 inhibitor. So as we increase the concentration of the drug, we see a decrease in BRD4 binding at its target gene. So we can clearly see these differences in the ChIP DNA. So it must be a problem in processing the samples for library generation and sequencing. So at what steps might we be losing this difference? The first would be during library generation. Library generation requires PCR amplification. And for us, we do 15 to 18 cycles of PCR to get enough library to sequence. So you can imagine that during that amplification, you may be losing those differences between the two samples. The second opportunity is actually during cluster generation before sequencing. So if you're doing multiple samples with barcoding, it's common practice that when you prepare your flow cell, you want to have equal cluster density for each sample. So if there are quantitative differences in the amount of library, you will actually normalize them out during that step of cluster generation. And the last point is it could occur bioinformatically. It's also standard practice when doing multiple samples after the sequencing is complete to normalize those data sets to a common number of sequence tags. So for us, if, for example, if we're doing a 5-sample experiment, we might normalize all those samples so that there's 20 million sequence tags in each data set. So we started exploring ways that we might correct for this problem, and the approach we settled on was to use a spike-in control that is added equally to all the samples, and we could follow that spike-in through the procedure and normalize back to that spike-in at the end. There is precedence for using a spike-in strategy in a whole genome setting. In particular, this paper from Rick Young's group in 2012. They were looking at gene expression in normal cells and in cells overexpressing MYC. The cells that overexpress MYC have a dramatic upregulation of the entire transcriptional profile of the cell. So the expectation is that everything in those cells are being upregulated. And in fact, those cells that overexpress MYC have 2- to 3-fold more RNA than a normal cell. When they did the RNA-seq experiment, they actually saw that there was only a subset of the genes that were being upregulated. Many were not changing, and some were being downregulated. So they decided to use a spike-in strategy, and in their strategy, they used 92 synthetic polyadenylated transcripts that were added to the RNA prep. When they repeated the RNA-seq experiment with the spike-in and normalized back to the spike-in, they now see the expected result, which is everything is being transcriptionally upregulated. So the problem we're experiencing is not dissimilar to the problem I just described from the paper. And at its most basic level, that is that we're expecting global changes and we're not detecting those in our whole genome readout. So we also decided to use a spike-in strategy. We decided to use a small amount of Drosophila chromatin and spike that chromatin into our human ChIP-Seq reactions. Not only did we just spike in the Drosophila chromatin, we also added a second antibody. And this antibody is against the Drosophila-specific H3K27me3 variant. So the idea was that if we added this antibody, we could consistently pull out a small portion of the Drosophila genome for normalization. If we use this antibody in ChIP-Seq using only Drosophila chromatin, we get many thousands of peaks across the genome, showing that the antibody does work in ChIP-Seq. If we use that antibody with only human chromatin, we don't get any peaks, indicating that the antibody doesn't cross-react with human chromatin. So just to summarize what those reactions look like, in a single tube, we have human chromatin, the H3K27me3 antibody, a small amount of Drosophila chromatin, which is roughly equivalent to 1/40th of a human chromatin, and the H3K27me3 antibody. After we do the sequencing, we take that data and we map it both to the Drosophila genome and to the human genome. And if there are any differences in the number of tags across samples that are mapping to the Drosophila genome, we normalize those to make them even, and then we'll normalize the human data set by the same ratio. So when we incorporated that strategy, the first thing we saw was that we consistently saw more tags mapping to Drosophila in our H3K27 trimethyl IPs in the presence of the EZH2 inhibitors. So if we look at the Genentech samples, we have tags mapping to Drosophila from the DMSO sample, but significantly more tags mapping in the inhibitor-treated sample. And we saw the same thing in the Constellation experiment, so that we have tags mapping in the DMSO sample, but many more tags mapping to Drosophila, both at 4 days and 8 days of inhibitor treatment. The idea is that we're going to normalize these tag numbers so that they're equal, so we'll normalize down the tag count in the inhibitor-treated samples, and we'll normalize down the tag count in the human sample by the same ratio. We also used control antibodies in these reactions. For Genentech, one of the controls was H3K27me3 acetylation. I mentioned earlier that we actually would expect to see an increase in signal from those samples. If we map to Drosophila, we're actually getting fewer tags mapping in the inhibitor-treated sample. So we're going to normalize these up so that they're even, and we'll normalize up the human tag count as well. That's exactly what we would expect for this particular antibody. The control antibody in the Constellation samples was H3K27me3. This mark should not be changing, and we don't see significant changes in the tag counts across those samples. If we apply that normalization strategy to the samples, this is what the data looks like. So this is with the Genentech inhibitor. If we use our normal ChIP-Seq protocol, we don't detect differences in K27me3 between DMSO and inhibitor-treated cells. We also do not detect differences in the control antibody. However, after spike in normalization, we now see a dramatic reduction in the signal from K27me3 ChIP-Seqs in the presence of the inhibitor, with no significant effect on the H3K4 control antibody. So the first time we ran this experiment, we saw a 5-fold reduction in signal in the inhibitor-treated cell. The technical replicate showed a 4.5-fold reduction, and we just recently got back the biological replicate that also showed a 5-fold reduction. And this was right in line with what we were expecting based on Western blot data. To show that how dramatic this normalization is, now I've just zoomed in on a single location in the genome. Again, no major changes using the normal ChIP-Seq protocol. But after we incorporate globally, we've taken all the data and plotted it with box plots and scatter plots. For the scatter plot, each point represents a peak that was identified in the data set in the EZH2 inhibitor-treated sample versus the DMSO sample. And if these two samples are the same, that data would line up nicely along this diagonal. So you can see using the normal ChIP-Seq strategy, those samples are quite similar. After we incorporate the normalization, you see a dramatic reduction in the EZH2 inhibitor-treated cells. The H3K4 trimethylation antibody, we don't see a big difference between EZH2 and DMSO-treated cells, and the spike-in doesn't really affect that data. And for K27me3 acetyl, which we expect to go up, the spike-in actually does normalize that data up. So to summarize, by looking at the box plots, we see no difference using our standard protocol in K27me3 trimethyl, but a dramatic reduction with the spike-in strategy. The control antibody is not changing, and that also doesn't change in the presence of the spike-in. And we see no significant difference in K27me3 acetyl using our normal protocol, but a slight increase if we do the spike-in normalization. We applied the same strategy using the Genentech samples. Here we had DMSO-treated samples, 4 days of inhibitor and 8 days of inhibitor. Using our normal strategy, we see no differences in binding across those samples. After the spike-in, we see that we now have a dramatic reduction both at 4 days and 8 days. Again, if we look at that data globally with the scatter plots, we see the same effect, that without normalization, those EZH2 and DMS inhibitor and DMSO treated samples are very similar, and the spike-in dramatically reduces the signal from the EZH2 inhibitor samples. And that's true both for the 4-day and an 8-day of inhibitor. Our control antibody, which shouldn't be changing, again, is very similar between treatment and no treatment, and the spike-in doesn't change that data. And you can see that clearly again looking at the box plots. So what do we think is going on? I think the first thing is to note that there are differences depending on what antibody you use. When we do ChIP-Seq in the presence of a BRD4 inhibitor, we can clearly see the reduced signal in the presence of the inhibitor. So we don't know how globally this is going to be applied to different transcription factors or antibodies, but we do know that it's necessary when there's global changes for K27-trimethyl, and we seem to shift our data as expected with the K27 acetyl antibody as well. So what we think is going on is when you use an antibody against H3K27-trimethyl, that antibody is specifically pulling out the majority of the DNA in the reaction, and only a minority of the DNA is coming from background or from, in our case, the spike-in. So in the presence of the inhibitor, you're pulling out much less DNA, but the background and spike-in is remaining consistent. So in the end, you have more ChIP DNA in one sample versus the other sample. During the amplification, the one with less DNA becomes over-amplified, evening out that signal from K27-trimethyl. But during the over-amplification, you over-amplify, and you can see that over-amplification in the Drosophila spike-in. So if you normalize this out, you can recover those initial differences. For BRD4, we actually think that the majority of the ChIP reaction is background, and a minority of BRD4 specifically pulled down DNA makes up a minority of that reaction. So in this case, the amount of ChIP DNA coming out between DMSO and inhibitor-treated samples is equal. When you do the amplification, they now amplify equally, and you maintain that difference in the BRD4 samples, or in the inhibitor-treated sample. The last point I want to make before we wrap things up is that the Drosophila spike-in is not only limited to normalization of global differences. We've also used it to monitor the quality and consistency of our ChIP reactions. So there were a lot of steps in ChIP and a lot of opportunities for errors when processing the samples. And you can monitor the consistency using qPCR of Drosophila sites. So an example is in the data I'm presenting here. We did 3 separate experiments with K27-trimethylation antibody using the same chromatin. And so we had samples that were DMSO and again EZH2 inhibitor-treated cells. In the first 2 samples, we see the expected decrease in the presence of the inhibitor by qPCR. If we look at the Drosophila signal using Drosophila-specific primers, we see that we have a very even signal. So that shows us that there is consistency and no loss of sample in our ChIP reactions. So we see that in reaction 1 and in reaction 2. In reaction 3, we actually don't see the expected differences when we do K27-trimethylation ChIP followed by qPCR. And if we do the Drosophila qPCR, we don't see that these reactions are not even. So we had sample loss or some technical error in that first sample. So if we normalize these to become even and normalize the human data by the same ratio, we now see data that is very similar to the experiments that had good technical consistency. So I think if you're doing multiple sample reactions and you're expecting differences, this is a good mechanism to show that those differences are real and not due to errors in processing. Additionally, if you're going to do ChIP-Seq and you're going to make the investment in generating libraries and paying for sequencing, it's worthwhile to do these quality control checks up front before you move on to sequencing. In conclusion, what I've shown is that over the years we have not been able to detect global differences in K27-trimethylation by ChIP-Seq. So if we use standard protocols, we don't see these differences. And we've come up with a spike-in strategy that now allows us to consistently detect global changes in K27-trimethylation. And in the last slide, I showed you that a similar spike-in strategy can be used to actually monitor the quality and consistency of your ChIP reactions. And that can be done both for ChIP-Seq and in a ChIP qPCR setting. Lastly, I just want to thank people that were involved in the work. At Active Motif, a number of people from the services group who did all the ChIP reactions and all the spike-ins and contributed to bioinformatics. At Genentech, was involved in experimental design. Golfam helped with sample prep, and Sujit contributed to bioinformatics. And at Constellation, Patrick Choger was involved in experimental design, and others contributed to bioinformatics and sample preparation. So with that, I can take any questions.
Thanks, Dr. Egan. That was a great talk, and I will now open up the Q&A session. So to the audience, please submit any questions you might have. There's already been a few posed, so we'll get right into it. The very first one was, in slide number 13, what cells were you using in the Western blots?
Just because this is a collaborative effort with pharmaceutical companies, I can't really comment on details of the experiments in that way. So I'm not going to be able to get into cell types, drug concentrations, and those types of details.
Fair enough. Next question, why is Drosophila chromatin used? Why not some other organism?
So in theory, any organism could be used. We liked Drosophila, one, because it has a small genome, so we won't overwhelm the system with the spike in. And two, we were aware of this antibody that specifically recognized the Drosophila protein. With that, the system can become universal, that you can use the same antibody as with your spike in any system.
Sort of going along with that, the quantification of the human and Drosophila chromatin must be pretty important for this. How are you guys quantifying that?
I'm assuming that means how do we quantify the amount of chromatin going into each reaction?
Yeah.
So once we prepare the chromatin, we take out small aliquots of that chromatin, reverse the cross-links, precipitate the DNA, and quantitate the amount of DNA that's coming out. So it's actually not a quantitation of chromatin protein, but a quantitation of the DNA.
Is it possible to do Drosophila spike in the absence of the H2AB antibody?
In theory, it is possible. Any histone modification antibody will cross-react with those modifications in Drosophila. The reason we didn't do that was, one, we wanted to make our spike-in system universal so that you wouldn't have to start thinking about your spike-in, whether you're doing transcription factors or histones. The second reason was that we thought that if there were global changes occurring in your sample and you used the histone antibody, that you might effectively be changing the concentration of that antibody in each of those samples. So if there was dramatically more of the modification in one sample, you would have more antibody binding, and the other sample would have more free antibody. And that might actually affect the pull down of the Drosophila chromatin and artificially skew the normalization.
So the next talk starts off with a compliment. Great talk. Do you have a kit for the spike-in available?
We do not. So right now the antibody is available, so that can be purchased through our website. We are working on putting together the Drosophila chromatin and possibly primers for Drosophila as well, and packaging this into a kit.
So another kit-related question. Brilliant presentation and utterly informative. Another compliment. Can you actually apply the same approach for low-signal ChIP-Seq?
Yes, I think from what we see, the better the antibody that's targeting the human, the less spike in it comes out. So that means the opposite is the worse your antibody is against humans. So let's say the mark you're targeting, the antibody is not good or it's present at low levels, the more spike in tags that you'll get. So I think it's important that you don't overwhelm your system with spike-in, and I think there's going to be some variation on how much spike-in to use depending on what antibody you're using. But in theory, the answer is yes, it could work in any ChIP reaction.
If you think the over-amplification is due to library preparation, why not just add Drosophila DNA immediately before your library prep?
You know, we didn't go that route. We wanted our spike-in to mimic the complexity of the entire reaction, and we wanted to be able to follow it through the various steps. That was our starting point because that's what made the most sense for us. So we did not pursue spike-ins at other stages. This seemed to work so well that we didn't readdress whether it could be done a different way.
What ratio of human to Drosophila antibody are you using?
So the antibody is about 8 times higher in human versus the Drosophila antibody. And additionally, the amount of Drosophila chromatin, we're using 40 times more human chromatin compared to the Drosophila chromatin. And those are starting points that may be tweaked depending on the nature of the experiment.
A services-related question here. If you add in spike and control to a service, does that add cost? Does Active Motif add extra cost for that if they're doing ChIP-Seq with you?
No, our goal is to get people answers that are accurate, and if it takes spike-in to do that, that's just included in what we do.
Have you worked with any peak caller labs to have a patch that would allow you to run tools taking into account the spike-in aligned files? And if so, which tool did you use, Max or some other?
No, right now I'm not the bioinformaticists, but what I can say, it's all kind of done manually at this time. There are no tools available. We're mapping separately to the human genome. For K27, we use CISR. and we're mapping separately to the Drosophila genome, counting those tags and making any manipulations to tag counts in the human manually. But depending on how widespread this becomes or how many samples need this, it's something we would certainly be interested in developing to simplify the normalization.
Do you see a reduction in DNA yield after chipping for H3K27, meaning reduced DNA yield in the presence of EZH2 inhibitor, sorry?
Do you think that they're asking if there's less DNA coming out of the chip reaction?
Less when you use the inhibitor versus when you don't, the EZH2
Yes,
inhibitor.
so we quantified the amount of DNA coming out using a qubit, and we do see less chip DNA coming out of the reaction. It's very much in line with what we're seeing by Western blot and the results we've seen by the ChIP-Seq.
This person is a new entrant to ChIP-Seq, and they're asking, when you compare treated versus untreated samples, do you average sequencing information from the controls and under each condition?
Yeah, so the control in this case would be sequencing of an input sample, which is unprecipitated genomic DNA, and we do pool the input from both samples into a single control reaction, and they're both normalized to that same control.
How many reads do you think you lose to the Drosophila genome? And do you need to multiplex fewer samples for this technique?
In the studies with K27, the numbers have been somewhere between 500,000 and 5 million. So let's look at the one Genentech sample. I think we had 1.5 million Drosophila reads in the DMSO treated samples and somewhere close to 6 million in the inhibitor treated samples. So, if you're sequencing somewhere between 30 and 50 million reads per sample, losing 5 million is not going to dramatically affect your data. But as the person suggested, you can also then maybe increase the amount of sequencing you're doing to compensate for that. But those numbers can get skewed when you start looking at other things like transcription factors or less prevalent histone modifications. But for the experiments we presented here, it's somewhere between 500,000 and 5 million.
Can this spiking strategy be used for other ChIP-Seq, saying other than histone chip? I guess maybe transcription factors or something like that?
Yeah, absolutely. Now, we don't know that it's necessary to detect differences. As I said, we can detect differences in some of these transcription factors without the spike, but certainly can be used at the very least to monitor the quality and consistency of the chip reactions.
This person is asking, it says they read conflicting information about whether IgG or some other input sample needs to be sequenced for normalization as well. Have you guys considered that or looked at that versus your spike-in approach?
I don't like IgG and ChIP at all, actually. The first reason is that you're looking at a reaction completely unrelated to your specific chip reaction. So you're using an antibody that's different and trying to apply whatever happens in that reaction to your reaction of interest. In regards to sequencing input, the goal of sequencing input is, especially in cell lines, you can identify duplications in the genome. And those come across as appearing as real peaks. So it allows you to subtract out false peaks based on genomic duplications. For other reasons that we don't fully understand, you always get false peaks, and those often come through in the input as well. And the IgG, you're being somewhat selective in what you're pulling down, and it just doesn't translate as well for normalizing against the things we're trying to normalize out. So we don't use IgG as a control.
Is the antibody AM29715, I guess histone 2A.V, is that a monoclonal?
That is not a monoclonal. That is a polyclonal.
Okay. Is it possible that Drosophila would have different amplification properties than human or mouse or some other species, which might introduce a different variable into your normalization?
I don't think so. And honestly, even if there were biases in amplification, the way we're normalizing it doesn't matter, because we looked at trying to normalize to certain regions of the genome for Drosophila, or focusing in on background or on peaks. And in the end, we are just using a total number of tags that are mapping in each sample. So if we're getting 5 million unique alignments in Drosophila for one sample, we're going to normalize to 5 million in the other sample. And again, since every sample is getting the exact same amount of Drosophila chromatin, even with biases, they should be behaving equally.
Back to the starting material question. So if you were to ensure the same amount of starting material with and without EZH2 inhibitor, where less DNA is presumably being pulled down, do you think that would eliminate the artifactual overamplification, therefore ensuring that some amount of DNA is used in the control and inhibitor treated?
Mason, maybe you can rephrase that or read it again.
Yeah, I'm trying. The way I understand this question is just if you're ensuring that the same amount of starting material somehow between inhibitor treated and not, do you think that would eliminate the over amplification issue?
A little bit of a confusing question because the starting material, I'm assuming, means the amount of chromatin going into the ChIP reaction.
Well, I think they're talking about after treatment.
After the ChIP, what we're pulling out.
Yeah, and then going into sequencing. So not starting at your ChIP, but starting at sequencing. I think the idea here is that once you've treated it with the EZH2 inhibitor, that there's less, that reduces the amount of recovered DNA. So I think if you were to normalize that, would that fix your overamplification issue, do you think?
Yeah. So I think that's possible, but I think the thing about that is very little DNA comes out of a ChIP reaction. So for most antibodies, you're in a range that you can't reliably quantitate the amount of DNA coming out. For K27, the antibody's really good and the mark is abundant. So you're actually pulling out levels that you can get some kind of quantitation, but it's still on the lower end. So I don't think it's completely reliable. But assuming that those numbers are reliable, you could do that, but it's not going to apply to most ChIP reactions where you're using different antibodies. And then secondly, I guess the question is does that bias you in any way when you're trying to look at differential binding, and differential meaning on-off binding at select genomic locations that may be occurring within this global regulation?
So does the spike in results vary depending on the antibody used to pull down the Drosophila spike in, i.e. how specific to H2AV are the H3K27 results?
We've only used H2AV as the spike in antibody. If we use that antibody, the question is, can you find another antibody that's specific to Drosophila, right? I think that's probably...
and would it change the spike in results?
No, I don't think so. I think if you could find another protein or antibody that was bound to a protein that was present in Drosophila and it was specific for Drosophila, that you could do it with a different type of antibody.
This question is, what if I pooled multiple inhibitor-treated IP reactions to get a comparable amount of starting DNA relative to the control IP? Would that eliminate the need for a spike in?
Well, in that scenario, you would see presumably the same result, right? Because you'd be artificially eliminating the difference by adding more. And so there, I think you wouldn't see any change.
They wanted to get your read on a suggestion. So if you deduplicated sequencing reads, this might also help remove PCR over amplifications. It says, any thoughts? Do you have any thoughts on how that might work?
Yeah, so that's standard practice for us. We always eliminate duplicates in our ChIP data analysis. So, in a good experiment, we may throw out 1 million reads, and in an experiment that's not so great, we could be throwing out 15 million because they're duplicates. So in this case, duplicates were removed from both the human mapping and the Drosophila mapping.
That looks like it's pretty much covered. There's a few questions, but I think they've been covered already. So we'll go ahead and wrap it up. I will thank you for your time, Brian. And once again, I'd like to thank Brian for a great talk, lively discussion, quite a bit of questions this time around. And thanks again to our sponsor, Active Motif. We appreciate you taking your time to attend and listen in, and hope that you've enjoyed this webinar. A recording of the session will be published really soon on the video section of the Epigene website. There you can also view other webinars and interviews with thought leaders in epigenetics and non-coding RNA research. So until next time, good luck in your research, and goodbye from everybody here at Epigene.